Hi, I'm Stanley Maloy. We're here at the USA Science Festival on a beautiful October day on the Mall in Washington, D.C. This is a really exciting event. Hundreds of booths telling the public about science and the excitement of science. Thousands and thousands of people out here learning about science, from kids to adults. Really great event. We're going to highlight for you some of the really exciting booths that tell you something about microbiology, and hopefully you'll learn something about microbiology and enjoy hearing what other people have to say.
Science! S-C-I-E-N-C-E! Science!
Let's go to the ASM booth and learn something about how microbes are good for us. This is Neil, and Neil's going to tell us something about this exhibit here. Could you just tell us what's in all of these containers, Neil?
Okay, so we just started here with some mud from just about any source. I used a salt marsh as mine, but you can take it from anywhere. Pack that in a column with some nutrients on the bottom. Got some instructions there if you can see how to do it. You'll just pack that in there, seal it up a little bit so you don't get too many smells. This is how it starts out looking. That's what we start out with, just a black muck. And then if you expose this to light, you can put it in a windowsill or an artificial light source. Expose it to light, in about 8 weeks you can get something that looks like this, all these pretty colors in here. And that's all due to microbial activity. Okay, we can prove that if you make this a little experiment. This is one we set up, covered it with foil for 8 weeks. It's identical to this column, but you can see there's no colors in there. This is all due to different photosynthetic activity of different bacteria. We also have a complete oxygen gradient here, so we've got lots of oxygen on top, no oxygen down the bottom, but you can see there's lots of organisms that can live in the absence of oxygen. So this is a nice little introduction to the diversity of microorganisms and how diverse they are and different activities that they can have.
So what's this good for? Why do people care?
Well, this is a good introduction to the enormous diversity and kind of things microorganisms can do. They can live under all these different environments. It's also a good introduction to the fact that these organisms are so diverse they can live under so many different conditions. They might exist on other planets, for example. There's other exhibits here, I think, talking about life on other planets. The fact that these organisms can exist under these conditions on Earth is good evidence that microorganisms might exist elsewhere.
So that would prove that there is life on different planets?
No, we'd have to actually get some evidence from those planets that there's life there, but we know that organisms can exist in those conditions on this planet. And the fact that there's similar conditions on other planets indicates that life might exist there. Okay, we got a lot more work to figure out if it really does.
Thank you, Neil.
Okay.
So this is Elizabeth. She's from the University of Wisconsin, and she works on squid and microbe interactions. And she's going to tell us a little bit about what she has going on here. Can you tell us about the squid that we can see on the screen here?
Sure. So this is the Hawaiian bobtail squid. We collect them from Hawaii, and the reason we study them is because they harbor a symbiont, a bacterium, that glows. And it uses the light from the bacteria to escape being eaten by fish. And we study them to look at how bacteria and animals communicate with each other from both sides of the relationship.
So how do the bacteria keep the squid from being eaten?
So that's a great question. What happens when you're a squid is that they hunt at night. And so the moon will cast a shadow. And fish underneath the squid will look for that shadow to be able to find them and eat them. So it's in the squid's best interest not to be casting the shadow. And it uses the bacteria and the light that the bacteria produce to break up the shadow, to shine kind of, we call it a biological flashlight, a living flashlight, to shine it under them and break up the shadow so fish can't see them.
So how do squid have just this one type of bacteria that's important for them? How come other bacteria don't get in and mess up the whole process?
Well, that's actually an ongoing research question. What we know is that a lot of different bacteria can start to kind of get near the squid and start to try to get into the special organ that the squid has for the bacteria, but that at the end of the day, only one species and only certain strains of that one species can get in. And so there are a lot of potential compounds that might help to weed out some of the bacteria that don't fluoresce or that don't luminesce. And also, if there are bacteria inside the light organ that aren't making light, they get kicked out. So that's one of the great things about the system, is that there's a lot that we don't know. And so we're learning more about it every day, and it helps us to learn more about other animal and bacterial associations too.
So this is really a great example of how bacteria can help a bigger organism, an animal, survive.
Exactly. It's an absolutely wonderful system, and it's very easy to understand because the output is light, right? We have the squid, and when it's glowing, it's in a productive relationship. And in return, the bacteria get food from the squid. And so both partners benefit in a very real sense, and so it's a really rewarding thing to work on.
It's a great story. Thank you very much, Elizabeth. So now we're going to go next door to see the exhibit that Harvard put on, talking about microbes and the important role of microbes in making different types of food. So now we're with Sarah. Sarah is from the Microbial Sciences Initiative at Harvard, and their booth here is focused on the science of food, what microbes do to make food. Everything from bread to cider to xanthan gums. Now, xanthan gum, that's interesting. What is xanthan gum used for, and how do microbes influence xanthan gum?
Basically, especially xanthan gum is used in a lot of the food products that we have nowadays, like even ice cream, salad dressing. It's present in, you would be surprised with the amount of food products, and it's very simple to obtain. So basically what you need is a carbon source, and sugar in most of the cases, and plain sugar, and you just add the right bacterium, and it's Xanthomonas. It's famous for being considered a pest of plants. So it actually grows on plants, on the leaves. And the funny thing is that it's able to, by using the sugar, to make these long chains, these polymers, and that have a very particular texture. And that texture is very convenient for the food industry because it allows substances to be more viscous and to have a more appealing texture. So you can actually even try, and if you touch it, you feel different viscosity according to different concentration.
Maybe we can see some if there's a jar.
So like this is one of the examples that you can actually, just by shaking it a bit, you can already see that the viscosity is different and it's very slimy. But this is what makes many food products so attractive because
it doesn't look very attractive.
It doesn't, But if we imagine that this is, I don't know, like some jelly, and it just makes it creamier, softer. People actually appreciate it more because if you wouldn't add xanthan, then it would be much more liquid and it wouldn't serve the same purpose. So it also depends on your final desired product.
That's a great example of how microbes influence a lot of different food products. One of the other kinds of food products that you have exhibited here that's a little different is the cheese. And in that case, instead of one microbe making one particular product that's used in a lot of different things, there are many different microbes that give the unique flavors, right? Could you tell us something about the cheese?
Oh yes, and I can even show it to you. Monica, who's been explaining here, everybody about how cheese is different, even if the base is the same. So you have milk and you have microorganisms, but depending on the microorganisms microorganisms that you have a final product. And it's different not just in terms of texture, because you can even see that the one is more, it clumps a bit, the other one is a very smooth paste. but also in terms of color, because this one has a mold in it, not just bacteria, and especially in terms of smell, it's pretty different. And we use different microorganisms because they work differently in milk. They have their excretion products. The way they change milk, it's different from one bacterium to the other one. So then, if you even use a combination of several microorganisms, then you have a different product, and you can actually manipulate that, adjust according to the final product that you're interested in. And and cheese and also all dairy products like yogurt actually vary a lot with just the community, the microbial community that they have inside, like kefir and Greek yogurt, for example, vili. According to the country that they come from, you can totally see a different community of microorganisms. So they are all different, they are all living together, and even if they're all working on milk, they all give a different final product in terms of acidity, of texture, of smell. So it's pretty impressive.
Now, these things you're telling us about, these are products that humans have made for thousands of years, and in different places they make different types.
Yes, sir.
Nowadays there's a lot of discussion about genetically engineered organisms that could do one process a little differently than another. How is that different when we do it in the laboratory versus the thousands of years of harvesting the right bacteria in nature?
Well, I'm a microbiologist, so maybe I shouldn't say this, but we cannot copy nature. It has millions of years of experience, so how can we compete with that? Nature is wisest. It's the wisest scientist, I would say. But we cannot copy it. We cannot, I would even say, fully understand it sometimes, because it's very difficult to characterize some communities. For example, kefir. You come across with tens of different species of microorganisms. So it's very complicated, and it also changes a lot according to location. So I would say nature is ahead of us in a lot of things, including this, but we can use the knowledge that we have to improve certain products. It's a teamwork.
We have a lot to learn.
Definitely.
Thank you very much, Sarah, and thank you, Monica. Well, that's it for part 1 at the USA Science Festival. We'll have more exciting stuff for part 2, the USA Science Festival, on microbeworld.org.
For a treat. When you use yeast, it goes in bread, and people get fed. When you add yeast to water and flour, the yeast uses starch to give the cell power, and in the process it makes gases waste, but the gas makes the dough rise, and you get bread that tastes great. You're in for a treat. When you use yeast, it can be a great tool for making biofuels. Relying on oil is a problem we must solve. Scientists have yeast that make lots of ethanol. The ethanol fuel can be used to power cars, making the earth a greener place. Makes yeast the superstar.